Before you start your notes, you need to make sure that you've done today's journal question. So if you have not done that, press pause, go back to your journal sheet and the instructions on Canvas, and answer today's journal question, please. Okay, so we're starting our unit in Chapter 12 on DNA, which should hopefully be reviewed from middle school. So hopefully it's easy for you, but again, that doesn't mean you get to not pay attention. You still have to take in the information and make sure you're actively learning. So DNA has three things that it has to do. It has to be able to store information and copy information and then transmit information. So the two that we're going to focus on right now are storing information and copying that information. We'll get into transmitting information in our next unit on protein synthesis. So if I go too fast, the best part about having notes recorded is that you can pause it. So if I move to the next slide before you're ready, just go back a couple seconds, press pause, write down what you need to write down, and then press play again, and you can continue with the next slide. So Erwin Chargaff, he was a guy, obviously, who came up with the base pairing rule. So he noticed that the number of cytosines was equal to the number of guanines. Same thing with adenine and thymine. They were all equal to each other when he looked at any number of DNA samples. So what he concluded was that because those numbers are the same, then adenine must pair with thymine, since they're always found in equal numbers, and cytosine must pair with guanines in equal numbers. So no matter what species it was, that number of adenine and thymine, cytosine, and guanine can vary, but adenine always equaled thymine and cytosine always equaled guanine. So that's where we come up with our base pair rule. So on this picture, I just want you to write down that A pairs with T and C pairs with G. You don't need to know anything about the purines and pyrimidines, just ignore that. But I want you to write down that A pairs with T and C pairs with G. So a good way to remember that is you have apples in the tree, A pairs with T, and cars go in the garage. So C with G. Cars go in the garage, apples in the tree. A, T, C, G. Okay, so there are monomers to nucleic acids. DNA, remember, is an example of a nucleic acid, and their monomer is a nucleotide. And their structure, they have a phosphate group on the outside. Those are those little circles down there at the bottom of the screen. And then you have that connected to a deoxyribose sugar. That's the pentagon that's there in the middle. And then those are joined to the nitrogen base, whether it be adenine, thymine, cytosine, or guanine. It doesn't matter what order the base pairs are in. They're in any order, and that's what makes you the way that you are. So my base pairs are in a different order than your base pairs are, and that's why we look different. We make different proteins, and we don't look identical to each other. So again, going back to the base pair rule, we call these strands complementary strands. So if I give you, if you look at the picture, if I give you GATC, that means that the complementary base pair is CTAG, because A pairs with T and C pairs with G. So that's super important for you to remember. That's one of the easiest things we're going to do throughout this unit, is going through and writing the complementary base pair strand, because A always pairs with T and C always pairs with G. So that's one of the most important things you need to remember. Holding the two strands together, so looking at that same strand, holding them together are hydrogen bonds. And the best thing about hydrogen bonds is that they're very weak. So they're strong enough to hold them together, but when we need them to come apart, they're very easy to break, because we don't want it to be difficult to replicate your DNA. Your DNA is constantly being replicated. Think about all the cells in your body. They're all constantly dividing, which means we have to constantly be making DNA. So we need that bond to be weak so that it's very easy to replicate that DNA. So again, hydrogen bonds, best thing about them is that they're weak. It makes it easy for them to break apart so we can replicate the DNA. Bonding, we're actually going to skip this one. I don't want you to know, you don't need to know anything about the number of hydrogen bonds, so ignore this. Please don't write anything down. Okay, we're going to go through the replication process, and I do really want you to write every single thing on this paper down. Obviously not the picture, you have that picture in your book, but everything about the replication process, because you're going to need to know. So DNA is what we call semi-conservative, which means we're going to save half the DNA and put new DNA down on the other side. So we use one strand as a model for the new strand. So after the DNA has kind of unwound a little bit, helicase, which is an enzyme, it's going to unzip the strands, which means that it breaks the hydrogen bonds between those strands. So it creates a little hole. You can kind of see that down there in the picture. We have just a part of the DNA that has come apart. Then DNA polymerase, which is also an enzyme, is going to come through and lay down the new nitrogen bases. And it also does this cool thing where it can proofread the DNA strands. Of course, problems still happen, but it tries to prevent those problems and those mutations from happening. So again, in order, the helicase comes through and unzips the DNA strands, and then DNA polymerase comes through and lays down new nitrogen bases. It's very important that you remember what these two enzymes do. Don't get them confused with each other, because then you're going to become very confused yourself. So helicase, it unzips. Polymerase lays down new nitrogen bases. So let's kind of recap again. Helicase is going to come through and unzip the DNA, it breaks the hydrogen bonds. And then polymerase comes through, adds the new nitrogen bases according to the base pair rules. And DNA polymerase can also proofread each new strand. You obviously don't have to write that again. You just wrote it. You can if you want to, but you don't have to. So the telomeres, if you see anything that mentions the telomeres, those are just the very tips of the chromosomes. So they're kind of like caps on the end of the chromosome. They prevent the genes from kind of unwinding and breaking off and being lost. So that's really their only function. So what I want you to do now is you need to go to Edpuzzle, which there's a link on your Canvas assignment for today, and you need to log in and complete the DNA Edpuzzle. That's going to be your points for today. So let me actually just show you where you're going. I actually don't know that I have it on there yet. I don't have your assignment, but I am going to show you the page that your stuff is on. If we're not going to canvas.com, what am I doing? In structure, there it is. So right here, this is where you're going. The DNA structure and replication video on Edpuzzle. You might need to sign in. So if you go to sign in, make sure you go through your Google account, because we've already made these classes. These are for points too. So the DNA structure and replication one, you're getting points for this. So please make sure that you do it. I'm going to be checking that you've done them, and if you don't do it, you don't get points. So again, don't start off the second 9 weeks with a bad grade because you didn't do something. Make sure you answer all the questions on this video.